This meeting is being recorded.
Oh. Well, welcome everybody. Thank you all for joining. This is actually the 4th annual Ocean Acidification Researcher Update. And it's great to see so many of the members of the ocean acidification community online here today. I think just looking across all of these people just makes me think about the breadth and depth of our research community here in Alaska. And watching it grow has been such a cool thing. I'll introduce myself. I'm Darcy Dugan. I'm with the Alaska Ocean Observing System, and I'm the director of the Alaska Ocean Acidification Network. And I'm fortunate to get to work with all of you. This is, I guess I'll start off by saying thank you for that. And I'm coming today from the traditional homelands of the Dena'ina people in Anchorage. So today's session is going to be pretty much a fairly fast-paced who's doing what where in the ocean acidification world in Alaska, with the goals of trying to help strengthen our research community for collaborations and opportunities for data sharing, and trying to figure out if there's new connections that we can make. And speakers have 5 minutes, which I know is a very short time. And I've asked presenters to focus on recent activities, upcoming plans, and new results. But this is also a good time to bring up anything that you need help with, or anything you have to offer others, or additional resources that members of the community might be interested in, links or academic papers or upcoming events, or your talk or poster at the Marine Science Symposium. So please make use of the chat. Our rhythm is going to be a speaker will present. There'll be time for about 1 question per speaker, but realizing that there probably will be more questions. After you're finished speaking, check the chat, because we can continue dialogue on questions after your presentation as well over the chat feature. And if possible, if you have a question, ask it to everybody, because it's likely that others might benefit from the answer to that question. So before we get started, are there any questions on nuts and bolts for today? All right, we will proceed. I am going to share my screen because we're going to launch with some updates from the Ocean Acidification Network. And let's see. I'm going to ask you guys, are you looking at a PowerPoint of the network?
Yes.
Yes.
Okay. And that's all you don't see my other distracting things on my desktop.
No.
You're good.
Great. Okay. Well, the Ocean Acidification Network is actually celebrating its 5th year this year, its 5th birthday. And right now we have about 75 members of the research community that are involved either directly or tangentially in ocean acidification, or have asked to stay abreast of the latest research in with the communication with the researchers from the network. I've listed, so it's really cool to think about just over the past 5 years how much the research community has grown. And I've listed some of the primary goals that we started with in 2016 that I think still represent the goals that we're working towards right now. And I would say this past year, we have really been focusing on the first bullet of engaging with communities to expand understanding. And I'll be providing a couple stories from this past year on that front in just a minute. But really trying to build on both existing resources and expanding new ways that our network can engage with Alaskans. And I think one of the most important parts of this network is that it does extend beyond the research community, realizing that ocean acidification is very multidimensional in nature, and it's going to need everybody. And there's a lot of Alaskans that are either already engaging or are ready to engage. And we've been trying to put thought into how we create stakeholder researchers and engagement at multiple levels. And I know that a lot of you are already involved in these efforts and have really great ideas. And I also included the photos of the executive committee at the bottom right, because we've been asking a lot of them recently and really want to thank them for their time. I just wanted to note real quick a couple of resources that you may already be aware of that you can share with others. We updated the species impact poster this year for 2021. that's the one with the arrows, with some additional species since since it was first produced in 2017. The Kelly Lab updated the annotated bibliography of species response. So this is more detail than that poster that goes through study by study. And then I also just wanted to mention on the outreach front that we can work with you to try to get your research out into the world. So if you come up with a new article or paper, we can connect you with a radio journalist or a print journalist to try to help with circulation. So I'm hoping that you think of the network as made up of you and for you, and that you use it as a resource. And like the e-news for the Ocean Acidification Network currently has about 1,300 people. So with various arms of communication, there's a lot we can do. Let's see. I just wanted to make a couple more notes on engagement before going into a couple of specific projects. Listed here are just some of the audiences that we've been working with. Engagement with tribes is a key component of the network, and we're going to hear several updates today from members of our group here and from the regional programs. And I think one of the things there that the network can help with is working with, I guess, connecting researchers where appropriate, and also trying to think of how communication, how to format information on ocean acidification, how to bring the local level information back to the communities. And I know that regional programs are working together on this, and it will be exciting to hear some of those stories. We also continue to partner with the Salmon Habitat Information Program, which used to be part of the United Fishermen of Alaska. And they've been really great in helping circulate ocean acidification recent results or events, and also providing feedback on some of the outreach material that we produce. We're working to try to get a hearing during this last legislative session in Juneau, probably virtual. So we've reached out to committee chairs about what they would be willing to entertain. Also, working with the crabbing industry has become a really important arm of the network. I think there has for a long time been interest on the research side to make sure that the latest ocean acidification information was reaching a really critical industry expected to be impacted by changing ocean chemistry. And there's also interest from industry leadership. And we co-hosted a mini symposium in April that had ocean acidification researchers and crab researchers and crabbing industry leaders and crabbers. And I just want to give a shout out to Darren Pilcher and Esther Kennedy and Jessica Cross and Chris Long that were all part of that effort. And I think probably many people here have been following a pretty drastic decline in crab in the Bering Sea in some of the most lucrative fisheries. And the latest science has shown that we can't currently connect the declines to ocean acidification based on the information we have now, but it's really important to keep this dialogue going and build those relationships so that we can easily share information. And I just want to note that Jamie Goen, she's the executive director of the Alaska Bering Sea Crabbers, is on the executive committee of the network. And also Scott Goodman, who's the director of the Bering Sea Fisheries Research Foundation, is also on the committee. So it's really been great to have that. Just a note here, you've probably seen this before, but another plug for the new podcast called The Future Ocean. And this was produced by the network this fall, and it was in response to interest from the fishing industry on solutions. Because probably for all of us, when we present on ocean acidification, one of the most common questions is, well, what can we do? And so this is a 6-part series that introduces carbon policy, and it's informational in nature. It's not advocating for specific legislation, and it interviews experts, so scientists and economists and folks from the energy sector, and also some fishermen about how carbon policy works, what the terms mean, how it's being used other places. And so the goal here is to help create a more informed industry that can better track and weigh in on policy. So we hope that you've checked it out. If you haven't, please do, and share with your colleagues and friends. And I want to underscore here the uncountable number of hours of Dorothy Childers, who's on the executive committee of the network, that she put in all volunteer hours, and also Edie Mari, who is a student at UAF, who's been working with the network since September. another couple of quick things is we're getting a new website and it's going to be unrolled in February. I haven't actually shown this mock-up front page to anybody, so you guys are the first people to see it. It's going to have a more modern look and feel and also some updated information. So special sections on regional conditions and species response. And thanks for many of you who have contributed content and been in touch with Edie as we try to refine that content. And then that's an example page of like each species will have its own page that explains kind of in general what we know about it and some recent studies. And then you'll be able to click on those studies to see kind of bulleted. So someone who is curious, but maybe doesn't have a hard science background can still get the gist of what some of the research did. And we will be running these draft pages by you before they go public. So right now we're still working on content, working with the graphic designer, but keep an eye out for that. I should also note that we are trying to put a little more effort into social media. I have personally struggled to find both the time or expertise to do a good job with this, and Edie is helping set up a structure, and we're targeting February for 28 days of ocean acidification. So we would love to highlight what you're doing. Please send us ideas. I'll probably be reaching out to all of you with a little survey. We're curious about how much you all use social media, what you would like to see more of in your feed, potentially from the network, what you'd like to see less of, and also if you would be willing to do a takeover for the day, like on Twitter, like follow me to the lab kind of thing. So we have some ideas. We look forward to getting your feedback on that. And then I also want to mention that place-based imagery is really valuable. And so we are always looking for great pictures of Alaska, the environment, the people, the lab, and we will credit you. Let's see. This is my second to last slide. On a series that's going to be starting up this spring that the executive committee has been working on pretty hard. This is a 4-part series, and we hope everyone on this call will participate. The goals are to start with, for each of these topics, I guess we sent a survey around to both researchers and stakeholders to get a sense of what topics they would be most interested in. And these are the first 4, but potentially could have another series next fall or so. I guess it would be this fall. But the goal is to try to do a synthesis of the latest information and have some facilitated discussion, and then use breakout groups to allow stakeholders and researchers to explore some of these ideas in more depth. So we hope that people would come away with, we'd have more informed stakeholders and also a better understanding of stakeholder needs and ideas for new collaborations that could both that could advance both monitoring and engagement. So we'll probably be sending out a save the date here next week for these sessions, and they will span from March until early May. And this is my last slide. And this is ocean acidification at the Marine Science Symposium. And there's 10 sessions. And I truncated the titles of these talks just into general topics, but we'll probably be hearing a bit about all of these today. But I encourage you to go to the Alaska Marine Science Symposium website to check out some of these great talks and posters. And then lastly, if you have ideas from for the network, please reach out. If you'd like to see things done differently, or you have new ideas, we would really welcome feedback, because this network is very flexible in nature and we can be responsive. And we would like to meet the needs both of those that are collecting information and using the information. So we're trying to be value added on top of all of the work that all of you are doing. So with that, I think I will stop sharing and we will move to our first researcher update, which is Hongjie Wang on Arctic OA synthesis and the Saildrone. It looks like, there we go. Great.
Hi, everybody. I am not Hongjie. Unfortunately, she is not able to make it today. She recently received a faculty appointment at the University of Rhode Island and is living on East Coast time now. And this is prime time for taking care of her family. So she's asked me to step in and give this talk for her today. So if you have questions about the work that I'm presenting today that Hongjie has led herself, I encourage you to check out the paper's been published. You can find it at JGRO using the title here in the bottom left-hand corner. But it's work that Hongjie conducted in conjunction with another postdoc at the Woods Hole Oceanographic Institute, Hagen Lin. And Bob and I were just sort of along for the ride. So I encourage you to send questions. We're also grateful for the support for this project that came from the Arctic Research Program. Essentially, we were asking a key question. So do changes in sea ice in the Pacific Arctic impact carbon cycling? As many of you may know, increases in light are hypothesized to increase primary production in the Arctic, which could increase the amount of carbon that is sequestered. Extra wind mixing might provide more nutrients, which could also increase production. But new stratification caused by salinity decreases from ice melt and warming might limit the amount of nutrients in the surface ocean, which would decrease the size of the annual CO2 sink. There's also open questions about what the variability looks like in space, as well as in the timing of all of these different events. And then, you know, as we all know, acidification processes in the Arctic are complex. They don't just happen from the surface ocean. We're also preconditioning the Arctic with acidified water laterally as well. So essentially what we did was create a time series for this geographic region using SOCAT and some recently collected Saildrone data. And that time series really only gives us enough data to work with during the summer. As we all know, Arctic winter data is extremely hard to come by. So I want to emphasize that what I'm about to show you doesn't represent an annual average or even a seasonal average, given that the seasonal cycles between the Bering Sea and the Chukchi Sea, and we looked at both, are different. So sort of think about this in your brain as June to October. So this is really the primary production season in the Chukchi, which doesn't necessarily line up with the Bering Sea all that well. Essentially what we found out is, you know, once we collected all of this CO2 data together, we combined it with a reanalysis wind product to create an annual CO2 flux product, which you're seeing here on the left. Warm colors are CO2 efflux and cool colors are CO2 influx, where the ocean is absorbing carbon dioxide. And this really helped us answer our first question: where is carbon entering and exiting the ocean? So those of you that are familiar with the region, as we all are, will probably notice that this looks very much like a water mass map. And that's what this plot on the right here is showing. We're using these violin plots to identify the carbon signature of different water masses. So CO2 flux in river water is close to zero, whereas these other water masses are all absorbing carbon at varying rates. So our second question was whether or not these processes are changing over time. And overall, what we found is that warming, perhaps associated with stratification, might be playing a role here. Overall, the Chukchi Sea carbon sink is decreasing. Remember that stratification mechanism I talked about before, while the Bering Sea sink over this time period is increasing. And we think that this increasing sink in the Bering Sea is due to primary production again over this June to October period. So this isn't necessarily an increase in the spring bloom, but due to increasing strength and possibly frequency of fall blooms. The frequency question is sort of open-ended for the community right now. We also included a section in this paper about differences in wind mixing between the front and end of this time series between the two blocks that we use to make the difference plot that you're looking at right now, showing that there is an increasing amount of upwelling winds in the later part of the time series that could be lifting nutrients up to the surface layer and maybe helping to contribute to that primary productivity in the Bering Sea. That being said, subdecadal swings and noise in the Bering Sea primary production cycle make it really hard to derive long-term trends. So all of that is broadly speculative. So there's lots of questions that this work has opened up for us. First of all, we don't have the data to really draw conclusions about changing salinity or water masses over time. So ice melt itself can have a really strong impact on carbon chemistry, but we need more high resolution data to specifically link salinity changes to changes in carbon chemistry, which is why you didn't see me talk about that in this presentation today. As always, we'd love to collect more winter data. This will also help us validate biogeochemical historical model outputs during the winter season so that we can otherwise use models to help fill an annual gap and get an annual perspective. And last, we want to continue to investigate the complex ways that the Arctic is acidifying, both from above and interface with the atmosphere, as well as the ways that preconditioned acidified water is transported to the Arctic halocline laterally. So I think that does it for me. Did I make time, Darcy?
Nice job, Jess. And maybe you can answer a question while Darren's getting queued up. And I just wanted to actually just ask you, I've got Hongjie's email here. Do you think she would feel comfortable with me putting in the chat?
Absolutely. It was on one
Okay.
of her slides, hwang.uri.edu.
Oh yeah, right. I'll stick it right there too. And thanks, Natalie, for sticking that paper in there. Any questions for Jess? Okay. I meant to mention at the beginning, we're starting in the Arctic and we're moving southward around the coast, starting with conditions. So we are now going to hear from Darren Pilcher.
Hey, thanks, Darcy. Can you see this okay? Can you hear me?
Looks great.
Awesome. Yeah, so for my update, or I should say first, I'm Darren Pilcher. For those who don't know me, I'm a research scientist with CICOES in the University of Washington. And for my update, I thought I'd chat a little bit about some of the work I've been doing for incorporating model outputs of OA into the Bering Sea fisheries management pipeline. I'm going to be focusing more on our annual kind of shorter term work. We also are doing kind of longer term multi-decadal projections. And if you are interested in learning more about that or my present talk, I would please direct you to my pre-recorded talk for AMSS in the Bering Sea section. So sort of over the years, we've been developing this Bering Sea ocean model with carbonate chemistry to kind of help with some of those challenges that Jessica described with observing in Arctic waters. So here I'm showing the bottom water omega, the aragonite saturation state for just the Bering Sea management area. That's kind of why it's sort of truncated off in different areas. And on the left is the average July to September conditions over the full model simulation. And on the right is the 2021 anomaly compared to that average. So sort of our plan right now is we've been updating this about twice a year, usually in May and then in September, to kind of keep this model run and this hindcast going. So for last year, the two noteworthy items were that the bottom waters near St. Lawrence, shown in those blue colors, were relatively higher than normal, whereas the bottom waters on the outer shelf were actually lower than normal, which is part of a multi-year pattern actually that first emerged in 2018, and which we think is likely caused by an intrusion of off-shelf water from the Bering Sea basin onto the outer shelf. That water is very old and has a very high carbon concentration, so it's sort of generating this local signal of really low omega values here on the outer shelf. Oops. We also use this information to develop index values for the entire shelf based on biologically relevant thresholds. So sort of like something like sea ice extent, we're trying to develop here this index that is kind of a one-stop shop to illustrate what the water conditions were like in the current year and how that compares to both the previous year and sort of the longer term time frame. Here I'm showing sort of where we're at right now with that index, which is basically the percent of bottom waters for the entire shelf, where the aragonite saturation is less than 1, which is the gray line, and then also where a pH is less than 7.8, which is shown in the black line. That pH value is based on some red king crab experiments that Chris Long and others at the NOAA Kodiak lab have done, which have found negative impacts to condition and survival starting at pH values less than 7.8. So essentially how to read this plot is as that value goes up, conditions are generally more corrosive, more acidic, and then are better when it decreases. So overall, both indices have slightly improved since 2020, which you can see is that kind of slight dip there in the dashed line, and are actually very close to the 2003 to 2020 average. Both of these also peaked back in 2013, which is sort of the last year of the very cold conditions in the Bering Sea. and have improved since. So this is illustrating, right, that ocean acidification is this gradual long-term process, and that there's going to be a lot of year-to-year natural variability in the meantime. I'll just lastly note that we're also updating this hindcast so that it starts in 1970. That'll be done actually later this week. And so we'll have a much larger historical perspective for this. And so with the help of our colleagues, particularly Elizabeth Sidden at the Alaska Fisheries Science Center, who graciously provided me with this slide, we've been incorporating these model outputs into the annual ecosystem status report. This started with a noteworthy section in 2020, where we received a lot of great feedback from the community and the SSC of the Fisheries Management Council, who acknowledged the usefulness of these products. You can see this quote in the center of the screen there. But sort of noted that there seemed to be little that they could do to help fix the problem in the meantime. And so in 2021, we expanded our index to include pH and also transitioned it into a full contribution to the ESR, with the idea being that we will continually update this on an annual timeframe, showing, for instance, those previous plots that I showed. So, and with that, I think I will stop.
Great, thanks, Darren. I should mention too that Darren has presented directly both to fishermen and crabbers in 2021. And I think there was particular interest. He had a graph that was looking at the threshold important to crab and how that would change under IPCC emission scenarios, which I think was a really nice way to depict how this data is important for future decision making. Great, Natalie. I guess real quick, any questions for Darren before we move into Natalie's presentation? Okay, feel free to put them in the chat if you come up with one later. And Natalie, we'll turn it over to you.
Okay, you can see my slide.
Yes, looks beautiful.
Okay, great. Hey, everybody. It's great to virtually see everyone. Hope to see everybody in person in the future. Darcy and the rest of the network, I just wanted to say thanks so much for this meeting and all the work that you've been doing for the last 5 years. It's just been so exciting to see how many more groups are not only monitoring, but are engaged, are teaching, are involved in the process and using some of the resources that are coming out of the Ocean Acidification Research Center. This is the lab that I lead at UAF. And here I'm showing a photograph of members of the Seward National Ocean Science Bowl team. So I've been meeting with them every few weeks. I haven't talked to them recently. They're getting ready to have their competition next month, but they're really interested in ocean acidification. Climate change was the theme for this year's National Ocean Science Bowl. We've been going through a lot of the resources on the network website. They kind of started out really interested in monitoring and some of the applications. I think most recently, they've been really excited about learning more about CDR, carbon dioxide removal. So that's been exciting. They were able to go out to some of the sites that I have been working on monitoring in the Gulf of Alaska in November. So this is a picture of them on the RV Nanook, and they got to collect samples and see data and be a part of the whole process. So it's just really exciting that we have the network and all of the resources to kind of help train and educate and include new folks. So just to give you a really quick update, I do have a poster in the Bering Sea session. I really wanted to highlight the mooring in the Bering Sea at mooring site M2. This is a long-term mooring site that has been collecting data for over 25 years. It's led by the EcoFoci program. Phyllis Stabenow is the main PI on this mooring. I started monitoring carbonate parameters at M2 in 2011. This is a photo of it. It's lovingly called Peggy, after Peggy Dyson, the voice of the North Pacific, which many of you may know. So here I'm showing our data. This is month. Typically, this is a seasonal deployment where it's only out during the ice free season when we have a ship that can go out and get the surface buoy in and out. This has been an anomalous year. It's still out there. So we're collecting data in these winter months. So that's something that you might have heard in Justin Hongjie's presentation at the beginning. It's a reoccurring theme. We don't have a lot of observations in the winter. So this is really exciting. So when you start to see a lot of these researchers, including some new monitoring, these data will be in their projections and summaries and analysis in the future. In October, we had a really big storm come through, bringing deep water up to the surface, and we saw we had a little bit of outgassing. We're seeing that again, assuming again it's from strong winds and storms. The sea ice is very close, so we're really hoping to get this out of the water. The ship was supposed to leave this morning out of Dutch Harbor. This is the 4th attempt to recover it since September. They have been delayed. Hopefully they'll be going tomorrow, but I've been very anxious, and there's been a lot of satellite calls in the last 72 hours, so we'll see. I hope I have good news in another couple of days. But I also just wanted to highlight a project that the Ocean Acidification Research Center started last year. It's called the Samples of Opportunity Program. This is available for current UAF students to add ocean acidification measurements to their research projects. The proposals are due March 15th. So if you're a student, you know a student, you want to get students involved in your monitoring or in your region, contact me and we can try to hook you up with someone at UAF if you're not already collaborating with someone. I started this last year, and one of the students that we funded to collect samples was doing a co-monitoring project at a mariculture site in Juneau, and it was a huge success, and it just showed how adding a new tool to your toolbox can be helpful, not only for all of us who are already monitoring for ocean acidification, but how can co-collection of various variables add to everybody. So if you want to contact me about this, or please spread the word, and I'll include the link to the site and the flyer in the chat.
Fantastic. Thanks, Natalie. That Samples of Opportunity seems like a really fantastic way to help students get experience and also contribute to the science community. So we will continue circulating that through the network as well. And that's also really exciting about the Seward Ocean Science Ball team. Do you know if their presentation at the competition is publicly accessible or will be recorded?
I will ask, and I'll let you know, and you can maybe let folks know through the network. Yeah.
Great. While Andie gets queued up, are there any questions for Natalie? All right, great. Let's see, Andie, are you ready to present?
Yeah, let's see if the technology will agree with me here today. Is that visible?
It looks great.
Awesome. Okay. Hello, everybody. My name is Andie Wall. I am the environmental coordinator at the Kodiak Area Native Association, also known as KANA for short term. I'm here to talk a little bit about the Kodiak community sampling efforts that are in collaboration with Kodiak tribes, KANA, and NOAA, and you'll hear a little bit more from Chris Long about other work that he is doing, but he's also a key partner in this project. For reference, this is the island of Kodiak that you see. There are 4 tribes off the Kodiak road system that are participating in ocean acidification sampling, in addition to sampling on the road system, actually in the city of Kodiak that you see right here. And so how it works is KANA is, and I act as kind of the liaison between the tribes and NOAA. NOAA collects individual samples, has a percolator. Kodiak tribes collect sampling actually weekly, and then those samples are shipped off to be analyzed at the Kodiak facility. And sampling started in 2019. It's been a little bit rocky getting started just because of staff turnover and because of, obviously, for COVID reasons. but we're taking surface and subsurface samples at all of these locations, which is really exciting. There it is. Okay, so changes from last year. What have we been working on to kind of revitalize the program and to kind of work on some issues that were identified since the program started? Because yes, it's very exciting that this program is ongoing, but we realized that there's a lot of tribal community efforts that are happening around the state of Alaska that don't really have a lot of collaboration in terms of how samples are being collected, how samples are being fixed, the data sharing, and the data sharing and how that information is being communicated to entities that are actually collecting the data. And so that's what we've been working a lot on. We've been working a lot on our quality assurance, reviewing the data to make sure that it's usable and accurate, standardizing protocols. And one example of that is Kodiak before this last year wasn't collecting samples to look for human error. And so we added 3 surface samples monthly for that analysis. We're also looking at data sharing, like I already mentioned. How are we taking this really important data and the concepts of ocean acidification and putting it into digestible information that tribes and communities can use to understand the impacts of ocean acidification in their own community? Is that something that we standardize reporting? Is that something that we, how do we create that content? And so that's something we've been working really hard on, and I'll share more examples on that in the next slide. But we've also been increasing collaboration with other networks and entities across the state. I think this has been key, and we're moving in a really great direction. Working with the Chugach Natural Resource Commission and Sitka Tribe to try and standardize the way that we are reporting the information that's being collected from these tribes. We've also run into some funding issues. This program is funded part through the Indian General Affairs program and partly through NOAA grant funding. And we need a new funding source to be able to continue this really important work. So our team has been working hard to submit grants to continue and expand on the work. Sort of the new outreach materials that we have been creating. I'm going to actually start on number 3 here, and because I think this goes into the point I was just saying about data collection and making the information relatable and understandable for everybody that's collecting. And on the right-hand side of your screen, you'll see something that our team has been working really hard on. It's a brief. It started as a 2-pager originally and has become kind of more of a 5-pager at this point in time. But the idea behind this brief is to explain what is ocean acidification, give a little bit of information about the impacts of OA on Alaska. This section has monitoring in Kodiak and an explanation, and then what parameters do tribal environmental samplers help monitor. And the idea is that this brief will go in front of all future reports. And so anytime tribal environmental samplers get a report on their data and all the samples that they've collected, they don't have to go back and research, okay, what does this parameter mean? What's the definition of CO2 and how does it impact these different species? And how does it impact our region? They'll have that information in front of them and before they even get to their data. And so that's something that we're really proud of and working on it. The next steps is standardizing reporting and looking at what information is going to be most valuable for tribal communities to receive in terms of their information and interpretations of that data. What we're also working on is the 4th annual Marine Water Quality Workshop that is going to be happening on February 28th to March 1st of this year. And that's an opportunity for Kodiak tribes to get together and talk about their individual sampling efforts and talk to other researchers across the state about their efforts. And one of the exciting things that's going to come from that is Switgard Justelo is going to create an Ocean Acidification 101 video that hopefully we can share with other entities around the state that just gives a brief description of what is OA. because we feel like we're kind of repeating ourselves a lot, and it would be great to have a resource available that explains that, that's local to Kodiak. So lots of cool, exciting outreach materials that are going on, and lots that we're working on, and hopefully this document, the brief, will be available within the next few months here. Thank you for the opportunity to share what we're working on.
Great, thank you, Andie. And I really want to applaud the efforts of KANA in figuring out how to report back. I think that's going to benefit the communities across Alaska and other regions too. So thank you for that. We look forward to seeing what comes out. Any questions for Andie while Jacqueline gets queued up? Jacqueline, are you on?
I am here. I'm just gonna start to share my screen. Can you all hear me and see that?
Yes, we can see your screen. It's not in presenter mode. So if you, there we go. Looks good.
Okay, great. Hi, I'm Jacqueline Ramsay. I'm the director.
Uh-oh, Jacqueline. It sounds like your connection may be a little bit delayed. Am I the only one having trouble hearing Jacqueline?
Nope.
No,
You're not the only one.
Okay, I'm gonna write a note in the chat here. Having trouble hearing you. If we can't get Jacqueline back, then maybe we'll just advance another presentation and see if she can reconnect with a better, more clear connection. Steve, are you on deck here? Steve Baird? Oh, looks like Jacqueline did drop.
Hello,
Anyone?
I'm sorry, I lost you all, or you lost me. I'll hope that this continues to work for me.
Sure.
So-
do you want to give it another shot, Jacqueline?
Sure, I'll try again here. So here we have a long-term data set. We're still working on
Real quick, Jacqueline, we don't see your screen anymore, so you might have to reshare that. Did you catch that?
yeah.
Oh yeah, great. Looks good. There, it's starting to go.
Okay. So on the left is our long-term data set for continuous monitoring through the HACI Institute. They developed the graphs, and we're working on the later years. Darn it. Sorry about that.
And Jacqueline, could you introduce yourself real quick? I don't think we heard it the first time because of the.
So I'm Jacqueline Ramsay at the Ocean Acidification Research Laboratory in Seward at the Aleutic Pride Marine Institute. And sorry about the problems here with getting going. I wanted to let you know that just a brief update on our monitoring efforts for continuous monitoring are ongoing. And we also process samples collected on a weekly basis by citizen scientists in our community of the Chugach region. We have 7 villages in our region, and we also have partners in Prince William Sound Science Center for the cruises. Rob Campbell's takes our samples there, and then the Ketchikan Bay Research Reserve cruises. And so with our villages, I should probably tell you it's King Cove. Outside of our Cirque region, we have King Cove, Seldovia, Nome, Ukiagvik, Kotzebue, and Little Diomede just came on board not too long ago. So we have a few samples from there. And our villages are we started our program with a grant from the BIA to start monitoring our community sampling program in order to get a baseline DIC profile for each village. We're hoping to get long-term data sets such as the one you see here on the right for Seldovia, which is a 4-year time series. And what we will have, let's see, that was, we have a couple more years that we have to add on to that as well. But what you can see is the aragonite saturation in orange and over the years, and you can see the pattern of saturation, which is what we're looking at for shellfish restoration sites and to get a baseline monitoring long-term data set from all of those sites. With the percolator and the improved capacity that we have for OA research, we have been getting into a lot of collaborations, and here's some recent collaborations that we've had. We've collaborated with Marina and Amanda over the years a few times now, and we're able to validate the manipulation, the DIC manipulation for species vulnerability assessments and species responses to changes in dissolved in organic chemistry. So what we try to do is look at ambient and then projections into the future for possible OA increases in Alaska. So far, this isn't a complete list, but we have looked at pink salmon, little neck clams, cockles, and done a little bit with kelp. Down here in the bottom, you see Amanda. She's got her setup going with kelp there. And at the top is Marina when they were here in the spring doing their juvenile pink salmon processes. We're also testing and validating instrumentation. Most recently, we've got a PCO2 to go handheld unit for the field and hatchery uses. There's a computer app that goes along with it so that you can put in what you're starting with and your targets, and you can provide the carbonate chemicals to the tank in order to manipulate it. We're also with the Ocean Foundation. This was all in collaboration with Burke Hill's Dacunalytics and the Ocean Foundation. And we're currently producing a manual for the PC2Go handheld unit. In the past, we've also hosted sunburst instruments for Wiley Evans, and we've done autonomous ocean gliders that we've hosted here in the hatchery through UAF. And we'd like to continue with these collaborations and increase them. And then as Andie and so many people have mentioned, our next steps are to improve community sampling and data sharing efforts. That's huge, and that's taking quite a bit of time in order to just make sure that the data is in a good report ready vehicle to give to our villages. We'd like to increase research collaboration at APMI. We could use more graduate student research. And I just wanted to plug the posters and presentations that are coming up at AMS this week. And that's about all I have for our ORLab update.
Great, thanks, Jacqueline. It's been a big year for Alutiiq Pride, and she mentioned several of those studies. Marina is going to be talking about a study here in a couple minutes that took place at the lab where Jacqueline is. Any questions for Jacqueline while Steve is getting queued up? Okay, you can always add some to the chat. Is Steve Baird on, or anybody else that's planning to present from Kachemak Bay? Chris Holderreed? No, okay. Oh, there's
Yeah,
Chris.
Darcy, I'm on, but Steve was going to present. I don't know if he's got issues.
Okay, well, we can always circle back in a few minutes and see if he has joined us. Does that sound like a good plan? I don't want to, if you have put anyone on the spot who is not prepared to present. So maybe we'll circle back to Steve in a couple of minutes. How about Chris Whitehead with CTOR? Chris, are you on? I know there was a possibility of Chris not being on, so he sent me a few bullets that I can share. So guys, one more call for Chris. Okay, not hearing Chris. His report included that, so Chris Whitehead is with the Sitka Tribe of Alaska and the CTOR program, Southeast Alaska Tribal Ocean Research. And he said they are hiring a new percolator operator. Their percolator system has been down since October, November, and that in the fall, new staff from the Sitka tribe went down to Oregon State University to train with Burke Hales on the percolator systems. And they're planning on having theirs back and operational by the end of January. And Sitka tribe right now has a backlog of community samples that have been collected across Southeast. And once their percolator is back online, they will start to work on that backlog. So in a second, I can add Chris's email to the chat if anybody is interested in reaching out to him directly. Is there anyone else from CTOR that would like to add anything else? Okay. And then I also, kind of along the community sampling Southeast vein, Wiley Evans from the CICOES Institute isn't able to join us today either. And the update from him was that the ferry Columbia, that's the Alaska Marine Highway Service vessel that travels between Bellingham and Skagway, which was instrumented several years ago with the CO2 monitoring system, has been out of operation, and they're hoping that that ferry will resume service in May. So fingers crossed for that. We had about 2 years of data before the ferry went offline due to budget cuts and some other issues, but it's looking hopeful that that will be back on the water. It collects ocean acidification parameters about every 3 minutes, I believe. So it's an incredible data set with both high temporal and geographic resolution. Wiley said his manuscript on the Alaska ferry analysis was accepted, so that should go into print here soon. And some other things he mentioned were both the Oceans Alaska and Sitka Tribe percolator records have been submitted to SOCAT for ingestion into the 2022 record. The Oceans Alaska percolator is located in Ketchikan. And he said that he will send the Sitka Tribe NCEI link when he has it. And in the meantime, he's got the Ocean Acidification Alaska data link. The Oceans Alaska percolator data link, which I'll put in the chat here in just a second. And he says, I'm working through the other sites to go to the NCEI database in the next month. And Burke and I, Burke Hales, are putting together a West Coast synthesis that needs to be submitted by May. So that's the report from Wiley Evans at the CICOES Institute. Let's see, and next we're going to move into species response. So we've got Shelby Bacus from UAF, who's going to be talking about the effects of multiple stressors on marine invertebrates. And great, Shelby, you've got your screen going.
All right. Hi, everyone. Thanks for joining. I'm going to leave my video off just because my Wi-Fi connection is not the best. But I'm Shelby. I'm a graduate student at UAF, and today I'm going to be discussing the effects of ocean warming and ocean acidification on the behavior and physiology of multiple marine invertebrate species. So for my first project, this took place in the Gulf of Alaska over at the NOAA UAF Kitsapina Bay Laboratory. And we looked at two species, the first one being Laudia scuta, which is the Pacific plate limpet, and its sea star predator, which is Avicerius trocheli, or the mottled star. And for this project, we really want to determine the individual, but also the interactive effects of OA and OW predicted for the year 2100 on 4 different parameters. We wanted to look at thermal tolerance, metabolism, cortisol expression, and the anti-predator behavior of Laudia. And our results were pretty interesting. I'll keep it brief and kind of look at the main points, but I want to start with the results from our thermal tolerance experiment. And what we found was that there was an effective temperature on the critical thermal maximum of Laudia. And we can see this in the upper right-hand figure. So we have CT max, or the temperature at which Laudia expire on the y-axis. And we have our different treatments on the x-axis. And what we found were that those individuals that were acclimated and raised in higher temperature conditions actually experienced an overall decrease in CT max compared to those acclimated to lower temperature conditions. And this is pretty interesting, and it's not what we hypothesized, although it's been also shown in other species. And we often see an increase in CT max for individuals acclimated to higher temperatures. And because we didn't see that, this possibly just indicates a lack of organismal thermal tolerance plasticity in the species, meaning that they might be more vulnerable to ocean warming and future ocean conditions. However, for some species that do show a similar pattern for this data, those species are often found in polar and subpolar environments. So there might be a correlation there and something to look into the future. And lastly, before I move on from thermal tolerance, I will point out too that the treatment that is showing the overall largest decrease in CT max is the treatment for the year 2100 scenario. And this also possibly indicates that the interactive effect of OA and OW make this species particularly vulnerable to ocean change in the future. Moving on to metabolism, we found that there was an effective pH and time on the mass-specific metabolic rates of Laudia. And we can see that in our figure on the bottom right as well. So on the y-axis, we have our mass-specific metabolic rates for Laudia, and on the x-axis, we have the time of our experiment in days. I will preface as well that we had a preliminary experiment looking at the metabolic rates of field-caught Laudia. So we had a comparison metabolic rate to the individuals acclimated throughout the experiment. And we did see that effect and that increase in metabolism over time and higher metabolic rates in the low pH treatment. Lastly, from this experiment, we found that there weren't any effects of pH or temperature on cortisol expression in Laudia, and that there wasn't an effect of temperature or pH on the anti-predator behavior of Laudia as well. Lastly, I do want to just discuss an upcoming project for this spring and going all the way through the following spring of 2023. I will be conducting an experiment on juvenile snow crab and tanner crab. We'll be doing a similar experiment where we look at the individual and interactive effects of OA and OW, but on a few more metrics. So we're going to look at morph metrics and survivorship, so looking at the growth and development of these two species in these different treatments, as well as physiological parameters such as thermal tolerance, metabolism, G expression, cortisol expression, and shell composition. And this project is going to take place at the NOAA Kodiak laboratory. So shifting from the waters around Kizitsna Bay and heading down to Kodiak. And that's all I have for you guys. So thanks for listening. If you have any questions, feel free to shout them out or you can put them in the chat. Thank you.
Thank you so much, Shelby. That's a really beautiful picture, by the way. We look forward to bringing you into the group that's been communicating with the crabbing industry as you launch this new crab project. Great. Let's see, I guess while Chris is loading his presentation here, are there any questions for Shelby? All right, Chris, on Kodiak Laboratory crab research.
Sweet. Can you all see my screen?
Looks good.
All right, excellent. So it's just an update as to what we've been up to the last year or so, and what we're hoping to accomplish in 2022, if things don't get worse. All right, so there we go. In 2021, we actually got a fair amount done, despite having a lot of limitations as to the number of people we could have in the laboratory. So in this, we get funded for kind of 3-year stints from the Ocean Acidification Program through its NOAA program. And really for the crabs in this go around, a lot of the emphasis has been in increasing the number of variables. So crossing experiments between ocean acidification and other factors. And then also trying to understand the mechanisms a little bit more. And that's looking a little bit more at the physiology as well as gene expression.
Uh-oh, Chris, we're starting to lose you a bit. Sounds like Chris's connection may be.
And so one of the first experiments, Juvenile red king crab. One of the crap. All right, I'm going to leave and rejoin.
Okay, we'll look for you to rejoin here in a second. I guess while Chris is rejoining, Marina, are you ready to jump in?
Yeah, for sure.
Okay.
Let me share my screen here.
Great. So Marina's going to talk about ocean acidification and juvenile pink salmon.
Okay, can you all see it in presentation mode?
Yes.
Awesome. Okay, so as Darcy said, my name is Marina Alcantar. I'm a graduate student in Amanda Kelly's lab at UAF. And I'm going to be just going through some results from the pink salmon OA study that we did this last summer at the Alutiiq Pride Marine Institute in Seward. And yeah, real quick, rapid fire here. Okay, so the figures you're looking at here, the top one, it represents conditional index of the fish over time. And this can be thought of as kind of like a proxy for fish quality. And then on the bottom here, we have mass over the course of the experiment within all of our treatment groups. And what we found for these two metrics was that there was a negative effect of pH on conditional index. So the fish that were reared in elevated PCO2 treatments had a reduced conditional index compared to those fish reared in ambient treatments. We also saw that there was a negative effect of pH on mass. So fish reared in that elevated PCO2 reduced pH treatment saw a reduction in overall mass. And we also saw one of food. So individuals reared in a reduced food availability environment saw a reduction in their overall mass. And I should say, we did this study to look at the combination of both ocean acidification and reduced food availability as both a direct stressor and an indirect stressor on juvenile pinks. I should have led with that. My apologies. So the next thing I'm going to show you guys real quick is looking at Vataratic Otoliths. And so this is really interesting because a question about the reason that Vataratic Otoliths occur in hatchery fish is something that's widely unknown. It's debated. So we wanted to look at whether or not elevated PCO2 can contribute to that. And so what we found overall was that within the study, we compared mortality fish to our sampled fish. And so that the mortality fish represent fish that died unexpectedly, not during our specific sampling time points. And so we compared the percentage of Vatarite within those fish's otoliths, the mort fish otoliths, to those that were sampled. And what we found was that fish that died unexpectedly, or mortality fish, had significantly more Vatarite present than fish that were sampled at our set time points. And so we're still investigating what this means, but it is really exciting that we potentially have, some kind of potentially an answer as to why we saw mortality throughout our experiment, and what Vatarite might do in terms of mortality. So our preliminary takeaways, we saw that exposure to elevated PCO2 or reduced pH resulted in a reduced conditional index, so a lower quality fish as well as a reduction in mass. We also saw that cortisol, which is an upcoming metric, so keep an eye out for that. Cortisol might be affected by the treatments. Overall, we saw that our results suggest in the future we may see smaller, lower quality fish under future OA conditions as a result of both direct and indirect effects of ocean acidification. And then whether or not there's an impact of Vataratic on mortality, or if this is just a contributing factor, or what it means for the fish overall is still under investigation. So our next steps include finishing up that cortisol analysis, gene expression, and doing an analysis of routine metabolic rate over the course of the experiment. And if you have any questions, feel free to ask or put them in the chat, or you can email me.
Great, thank you, Marina. And I should mention too that Marina is part of an interdisciplinary project looking at tipping points in the marine environment, particularly with respect to salmon, pink salmon, and our ability to predict those, and also look at the management structures in place to see how we can adapt our way through major tipping points with respect to ocean acidification and warming. And Jan Olberger, I know, was potentially going to make it today. Jan, are you on? He's part of the tipping points team and was the lead author on a paper that just came out in December that I can put in the chat here. It was looking at Prince Sound pink salmon and an assessment of data from the past 30 years and which factors were statistically significant in affecting the populations. And it was, according to the analysis, hatchery releases and temperature were statistically significant factors, but ocean acidification was not at this time based on the available data. However, realizing that may change going into the future. And I should say another component of that tipping points project is a fairly in-depth stakeholder component, and Molly Mayo, who I believe is on this call with the Meridian Institute, is in the process of interviewing fishermen, over 20 fishermen, on their thoughts and considerations of fishing, climate change management, and also ocean acidification. And that project will go for about another year, and then the team will be putting together the different parts, including results from Marina's assessment, kind of wrapping that into more finalized results that we'll share with everybody. Let's see, Chris
Darcy,
Long. Yeah.
Can I answer a question real quick that was in the chat?
Oh yeah, sorry, I missed that.
So Rich
Please.
Brenner, you asked if the fish were screened for disease. So we did throughout the experiment monitor the culture vessels and individuals for everything from oxygen saturation to ammonia, nitrate, nitrite. We also did bacterial cultures to make sure that we weren't seeing any kind of explosive growth. And then we did perform necropsies on mort fish to make sure that there wasn't any kind of signs of hemorrhaging or any sign of illness. And from what we've been able to distinguish, there wasn't. So they were very pampered fish. We're not totally sure why they were dying, but we're still working through it. Yeah, we also looked for signs of gas embolism due to the CO2 system, but there were none. And our levels were consistent with other experimentation that's been done, and it shouldn't have been a contributing factor.
Hey, great. Thank you. And really nice job. I learned a lot from that. Thanks.
Chris, are you back on? Okay, perhaps not. We'll circle back to Chris in a minute. Next would be Tom Hurst with NOAA on an update about groundfish.
All right. Are you seeing me now?
Yes.
Okay. Thanks, Darcy. Thanks, everybody. So I'm going to go over a couple of newer things on Alaskan groundfishes, one sort of experimental finding paper and one modeling and population level project. So last year we had a new paper about, on effects on walleye pollock. And this is a follow-up experiment to validate and explore some previous results we had a few years ago, but we wanted to make sure we were exploring all the possible impacts or not all of them, but more of them that we might see in walleye pollock. And so in one good thing, we didn't see any effects of acidification on the growth and survival of the walleye pollock larvae. And that's both good in itself because suggesting that pollock are somewhat resilient to that, but it's also good in that it was consistent with the observations that we had in the previous studies, suggesting that we can be pretty confident in that finding. But what we did find that we hadn't looked for before was that the exposure to high CO2 in the walleye pollock larvae reduced the rates of swim bladder inflation. And so the pictures here in this figure that you can see are fish with and without their inflated swim bladders from the experiment. And you can see the green arrows point to the presence of the swim bladder in fish in two different kinds of preparations of the biological sample. This top one is histology, and the lower one is a whole fish preparation. And then on the right, where the red arrows point to where the swim bladder would be in a fish of the same size and age, but is not actually present. And so we saw that more fish in the high CO2 experiment and treatments did not inflate their swim bladders. Now, swim bladder inflation is usually associated with higher mortality in fishes. So it's interesting that we didn't actually see that. So we saw there were no elevated mortality rates, but the fish didn't have their swim bladders. So the question that is lingering is whether there might be some delayed effects from later on in life of these fish not having their swim bladders. And it's our the physiological process is generally believed that if the fish don't inflate their swim bladders, they can't do it later in life. So if they don't do this, this is something they're stuck with for the rest of their life. So anyway, that's an update, and you can see the whole paper that came out last year. And so this sort of feeds into this broader perspective that we're building about understanding the sensitivity of different ground fishes. And so we put this little yellow here, adding the one more piece of evidence to our understanding of walleye pollock sensitivity. On another front, we had previously done work on northern rock sole OA sensitivity. And so that work was combined into a fishery population and stock assessment model to look at climate drivers in long-term projections of the model. And so this is a pretty complicated model, but the things that were included in this model are the OA effects on larval survival that we worked on in our laboratory, nursery temperature effects on the recruitment of northern rock sole, which is a phenomenon seen in the field, and then temperature effects on growth, which again is something we would see both in the field and lab. And these factors were applied in combination and individually. So, and just a real quick summary, one example of the stuff that came out of that, and probably the take-home message for this audience is in the model that had all three of these things, which in our paper is called Model 8, we saw a mean increase in the average recruitment over time because the negative effect of pH on larval survival was smaller than the positive effect of warming in the Bering Sea on the juvenile nursery habitats. And so we have to sort of think about in a big perspective how the different things might play off against each other. And so there's definitely still areas of uncertainty. The most important of which is probably how temperature and OA will alter the foraging opportunities and impact the fishery yield and profit. So there's still a lot of uncertainty, but we have a model framework that we can start incorporating these new effects into. And then just really quickly, what's coming up next? We just finished an experiment on Arctic cod exposing eggs to high CO2 and temperature variation. So I'll have that to present next year. I wasn't quite ready to show you that yet. And then we're about to undertake a second follow-up Pacific cod experiment, temperature and OA stress. And later in the year, we'll be doing the first OA sensitivity evaluation in yellowfin sole. And that's it for me for now. So thank you, Darcy.
Thanks so much, Tom. And I'm noticing a question here from Jessica Cross.
Yeah. Hey, Tom. One of the things that the community is really rallying around are these multi-generational and multi-life stage experiments. You were talking with the pollock about the lack of swim bladder. And I know for some of these larger fish, there can be logistical challenges with raising them across these life stages. And so I wondered, do you have an interest in doing in that work? Is it impossible to do that work?
Yes. So yes and yes, basically. Yes, we have an interest in doing that work. Yes, it is nearly impossible to do that work, but we do have a plan to start down that path using Arctic cod as our model species because the generation time is so much shorter. And so while we can't do multiple generations, we can look at the generational effect by exposing the parents and then looking at the effects on their offspring. And so doing multiple life stages, but whole life cycles for most of these ground fishes is not something that is just practical, and, given how long they live. So, but thanks for that.
Yeah. Thanks, Tom.
And thanks for that question, Jess. And Tom, I also want to thank you for your outreach recently, including a presentation on a lot of this to the United Fishermen of Alaska. There is definitely a lot going on in the species response world, which is really exciting. And I think you can keep an eye out for much of this on the new website, where there'll be a page for each of these species. I want to move next to Chris Holderreid. It sounds like Steve Baird won't be joining us, but Chris is able to give us a quick verbal update on some of the topics he was going to cover.
Yeah, thanks, Darcy. Yeah, it said apologies from Steve. And there was really just one thing he wanted to point out. He was going to show from the time series of the data from the water quality monitoring stations that we have in Kachemak Bay. So there's just for reminder, there's 2 stations, one at the Homer Harbor, one at the Seldovia Harbor, 2 sensors on each, one near surface, one near bottom. And those records are now over 20 years long. And so some of those long-term trends are starting to, be able to show up. And for kind of context of the work that Jacqueline and others were talking about earlier, Jacqueline in particular, at Seldovia with the aragonite saturation. Anyway, the long-term trends are showing that there's, slight but measurable decreases in the pH. So that's just a, interesting point that's coming out of some of the longer-term monitoring. So. Thanks, Darcy.
Thanks, Chris. And if Steve sends that graphic, we can circulate it with the follow-up from this call too.
Will do. Thanks.
Cool. The next presenter is Claudine Hari from UAF, but I know there is a chance she was maybe not going to make it, and I don't see her name here in the chat or in the participant list. So we may continue on, but I should note that Claudine has an oral presentation at AMSS. So I will point people there until we hear from her at our next convening. Let's see, next is the Regional Vulnerability Report Project, sorry. It was either going to be Tom or Kelsey. Tom, do you know which of you will be updating us today?
Yep, I'm here again.
Great.
All right, so you should be able to see this title. This is a new project that we're just starting, termed Evaluating OA Vulnerability and Interactions Among Traditional and Emerging Coastal Alaska Industries. It's got a very broad and wonderful team of researchers and from a number of different partner agencies and institutes you can see there. And so basically the goal of this regional vulnerability assessment is to look at a community level assessment of how ocean acidification is going to impact different aspects of the economy and well-being in different communities. And so you may be aware of this initial Alaska Community Risk Assessment that a number of us on the call were involved in. And that really focused primarily on the economic role of commercial fishing. And so this next assessment will broaden that out, include other industries such as shellfish and algae mariculture in sort of our assessment, and go beyond just the economic industries, economic uses to include aspects of community well-being. And so this project is going to focus on 4 communities, Kodiak, Homer, Sitka, and Hoonah. And as you all know, that in all those communities, commercial fishing and subsistence harvest and tourism are important, but all the communities have slightly unique characteristics that affect which industries are predominant in their area, how those industries share staff and facilities and resources, and interact with the community at large. So we want to look at those kinds of things. And so the way we will do this is to co-develop place-specific network models along with local community stakeholders to describe the linkages between industries and aspects of community and community well-being in these different communities. And then once we have those community supported models of their own internal dynamics, then we would apply an OA press perturbation to see how OA might alter some of these relationships in the communities. And then this is going to be an iterative process working with community members and industry representatives in the different areas. And then we'll have a separate model to, again, go back and reevaluate some of the economic valuation impacts. And so just really quickly about how we will do this, we'll start out using a qualitative network model approach. And it's an approach that describes the feedback linkages, the linkages and potential feedbacks between different nodes in a model, and includes in our case, we'll include the climate drivers, the sort of first level of marine dependent industries, and also the aspects of community well-being. These models are really flexible and adaptable to different kinds of nodes, and they can show how a perturbation in one aspect of the network ripples through and affects other aspects. So, and I should point out that the person that Darcy mentioned, Kelsey Furman, is the postdoc on the project, and she will be the one who's actually be doing most of the work for us. I'm, for now, sort of a spokesperson for the project and the lead PI. So anyway, thanks again, Darcy, and I'm happy to answer any questions if we still have another minute.
Thanks, Tom. And it looks like there is a question in the chat from Andie Wall, and she says, is there a cultural impact aspect of the surveys in addition to economic and harvest?
Yeah, thanks, Andie, for that. I'm hoping that we're going to be working with you and KANA when we do our work in Kodiak, and I know David mentioned that recently. So yes, in fact, that cultural aspect will be determined and described by the members of each community. So we will be asking them how they interpret the cultural impact of the marine resources and where they see that cultural impact playing and where the tightest associations of cultural impacts to marine resources are, so that we can capture, we can really do our best to capture how what we think is happening in the ocean would be playing out, to predict what the things that the community finds important and their self-determined aspects of cultural importance. Yeah, and thanks again, Andie, and I'll definitely look forward to talking with you.
Great. Thanks, Tom. I know that a lot of people have hoped for a next step from that 2015 risk assessment, so it's really exciting to see that move forward. Our next speaker is actually a member of the team that you just described. And at least I think that's next. Yes, next on the list, Marisa. Marisa, you'll have to help me with your pronunciation of your last name. I meant to ask you ahead of time, but Marisa is going to be sharing fishing community engagement and some work that she has been doing in addition to the project that Tom described. Marisa, are you there? I know she was on earlier.
She says she can't unmute.
Oh, shoot. Why don't we give Marisa a minute? And in the meantime, Jess, would you like to talk about carbon removal and give your carbon removal update?
Yeah, sure. I'm happy to jump in. Sorry, Amanda. I'll get back to our conversation in a second. Slideshow and share my screen. All right, am I good?
You're good.
All right, hi everybody. I wanted to offer a sort of update. There's been increasing interest in carbon removal in the Alaskan research community, and I wanted to talk really briefly about what that is for those of you who may be dancing around the edges of the field. So I know we're all carbon chemists on the line, so sorry if this is too much background for everybody here, but also to profile a proof of concept project that I'm contributing to, in that, you know, you all may have interest in. So for those of you who aren't necessarily familiar with carbon removal research, according to the IPCC special report on 1.5 degrees Celsius, stabilizing climate change at atmospheric temperatures even higher than those that we experience now is going to require negative emissions. Those are technologies that remove greenhouse gases from the Earth system. So this figure that I'm showing here from UNEP summarizes the need for negative emissions. So this is time on the x-axis and cumulative annual greenhouse gas emissions here on the Y. And so this yellow line indicates the projected business as usual greenhouse gas emissions, whereas the red line indicates the emissions limit that would allow for less than 2 degrees Celsius of climate warming. The shading in the background indicates the techniques that are going to be necessary for us to pull that yellow line down to the red line. Obviously, Plan A is conventional abatement technologies, such as a shift to renewable energy sources, which is shown here in the green shading. However, the IPCC acknowledges that it's going to be difficult to shift away from some fossil energy and other greenhouse gas emissions, like, for example, emissions from marine shipping and oceanographic work like ours. So to offset these stubborn annual emissions shown here in brown, we will need carbon removal technologies seen here in the blue shading. What was unique about SR 1.5, after having acknowledged this offset need for a while, is that we need to actually do more than offset those brown emissions. We're going to have to hit net negative. We need to actually remove legacy emissions from the atmosphere in order to meet our targets. So it's going to be extremely difficult to generate the negative emissions at the scales required here. The IPCC suggests that we're going to need to remove 20 gigatons of carbon dioxide per year from the atmosphere by 2100. Starting from year 0 in 2020, that's an approximate growth rate of about 6% per year. And by some metrics, that means we need to open hundreds of carbon removal facilities every year. However, at present, very few of those facilities actually exist, and most carbon removal technologies are not mature enough to operate at even a single gigaton scale. The biggest DAC plant in the world right now is about 1,000 times less than what we actually need. As we all know, much more research is necessary to understand how we can responsibly meet those targets. So if we're going to pull carbon out of the atmosphere, where are we going to put it? This figure right here is showing the 2021 global carbon budget that's put together by the Global Carbon Project. Naturally, the ocean sink is about 10 gigatons per year. The ocean can and does stably store a lot of carbon on the annual scale already. As we all know, that's a problem for things like ocean acidification, but it also performs an enormous global service by taking up all of that carbon dioxide. And there are a variety of ways that the ocean naturally takes up carbon. So when we think about how are we going to get carbon from the atmosphere into the ocean, how do we actually do that? So I'm profiling here 2 different methods. One of them is direct ocean capture. It's essentially the ocean's analog to direct air capture, where you run seawater across a membrane and you strip out the acid and you eject the basic solution back into the water. You can sort of combine that or think of that also as ocean alkalinity enhancement, where you're actually injecting a base into seawater in order to soak up some of that carbon dioxide. So the idea is to neutralize some of that carbonic acid. CO2 forms a weak acid as it dissolves in seawater. And so we want to really try to neutralize as much of that acid and store that carbon dioxide in mineral form, not necessarily as organic carbon, but in mineral form, where we can capture it on the order of about 1,000 years. but it's really, really hard to generate this alkalinity, which is why we want to pair it with things like direct ocean capture rather than mining that alkalinity off of the land. We ran a quick opportunistic project using Darren Pilcher's 10K model, which you saw profiled already this year, to check what the impact of an ocean alkalinity enhancement project would be in the Bering Sea. The project is actually being led by Hongjie again, Brendan Carter and Darren, among a handful of other supporting colleagues, which you see profiled here on this slide. Essentially a simulated addition of about 4 micromoles per kilogram per meter squared per second over 10 years was able to remove about 6.5 tons of carbon dioxide in total. And this is a higher uptake efficiency that's been observed in the other ocean basins, given that the Alaska region is naturally predisposed to soak up carbon dioxide for all the same reasons it's vulnerable to ocean acidification. It's cold, it's old, and the mixing works in our favor. I know that that's far, I want to emphasize that that's far from a ringing endorsement of ocean alkalinity enhancement. It's really just a start. There are a lot of open questions, you know, how are we going to get the alkalinity that we're using if we're not going to use direct ocean capture to generate it? Maybe we're going to be mining it. What substrate are we mining? That's a challenge. What do we do with the hydrochloric acid byproduct of the direct ocean capture process that actually generates that alkalinity if we're using direct ocean capture to remove acid and eject an alkaline product? We also need to use renewable energy to actually pump that additional alkalinity into the ocean and run that direct ocean capture process, or we eat up our potential carbon savings pretty fast. It's also essential to understand downstream as well as hyperlocal impacts. Those are key questions about the efficacy and the risks of this approach that tools like this regional model can't answer. We're gonna need some of these hyperlocal and global perspectives as well to assess methods like this. Essentially what that means is that CDR projects are gonna need to be nested. Last, it's really important to be able to observationally verify a project like this. We want to actually measure that it's having the impact that we said it would. And given the very small perturbations that the model identified that we might see in this surface seawater, it may be really difficult to use existing tools to be able to do this, or we're going to have to be very careful about where we take those measurements. So again, there's a ton of work to do before any potential deployment like this. So I'm looking forward to hearing from everybody else on the line. If you have other proof of concept CDR projects that you're working on, or ways that you think that we could work together to explore this new frontier of carbon research, I'm totally happy to hear from you.
Thanks, Jess. That is definitely underscoring the magnitude of our issue at hand. Are there questions for Jess?
I see a few in the chat here. Have I thought about harnessing tidal power? Yeah. As to whether I have like a specific plan, like, hey, we should use this tidal power facility to pair with this particular source. No, I have not gotten that far yet. I don't think anyone has gotten that far, but I know that there are some folks in Kodiak that are thinking about using the hydropower stations in Kodiak for things like offshore wind and things like that. How can we daisy chain some of these renewable energy sources together to power CDR projects? So it's certainly something that everybody thinks about, but I don't think anyone is anywhere close to proposing to deploy anything yet. It's all just a theoretical. Andie, I'm glad the background was helpful for you. Sometimes I feel like I'm talking down to folks. Anybody else?
Okay, I might direct further questions into the chat because we've had 2 other speakers that we've skipped over and are going to come back to. So thanks, Jess. Please add questions for her into the chat. And Marisa, are you connected?
Can you hear me?
Yes.
Hey, I have to send you my PowerPoint, Darcy. I don't know why I can't figure out how to unmute. Usually I blame the whole NOAA thing, but then Tom and Jessica figured it out along with a bunch of other NOAA folks. So. Can't do that.
I've got your, let's see. I'm opening up your PowerPoint right now, so I should be able to share on this end. Oops, let's see.
Yeah, it'll be pretty easy. Anyway, just to get things going, my name is Marisa Shimkoviak. I am a social scientist with NOAA, Alaska Fisheries Science Center. I am based out of Juneau. And as Darcy mentioned, I'm also on the project with Tom and Jessica and Kelsey and Dave and a whole lot of other people building those network models of ocean acidification impacts. But I think as many of you know, NOAA parses its funding mechanisms into ocean acidification and climate change. And so I also have a sort of broader climate change project that I've been working on over the last year that Darcy asked me to speak to. So that's what I'm gonna run through very quickly here. Next slide, Darcy. So the project is focusing on exchanges with commercial fishermen about their perceptions of climate change impacts on their fisheries and their capacity to adapt to that. In this brave new world of COVID, everything is happening virtually. So we've been doing interviews and I've had free workshops. This work is continuing in terms of getting more fishermen to talk to us about basically three overarching topics. What are the changes that they are seeing in their ecosystem, whether they're ascribing those to climate change or not, sort of a separate story. Responses to those changes, and then what do they need for framing their broader adaptation needs and resilience into the future. And so that graphic on the right just shows the communities that we've spoken to fishermen. So we're definitely recruiting for sort of more Gulf of Alaska community as well. Next slide, Darcy. So like this work is ongoing, and I'm not that big of a fan of presenting on ongoing work, especially in this really sort of highly sensitive and evolving area. But these, we've been having these exchanges virtually, and so we use Google Jamboards. And so this graphic on the right are just some of the ecosystem changes that fishermen are seeing in their system. I would say the most consistent and the ones that fishermen relate to having the greatest impacts are smaller fish, fewer fish, and ocean acidification. But really the discourse is around not necessarily how each of those results in impacts on their well-being, but the cumulative impacts of all those stressors. And more broadly also how uncertainty in the system due to climate change is impacting their well-being and capacity to plan into the future. Since this is an OA group, I will note, again, OA is one of the big impacts, the big stressors that fishermen note in relation to ecological changes in their system. It's parsed in their discourse from broader climate change impacts. So even for those who sort of express uncertainty that climate change is happening or express uncertainty about what climate, sort of where climate change is happening and how, they still believe in ocean acidification, which I think is very interesting as a social scientist. OA is also not specifically tied necessarily to ecosystem changes in the discourse for fishermen. So they're not necessarily attributing OA to other things that they're seeing, like smaller fish or algae or whatnot. Although they do relay concerns about food web impacts from ocean acidification and associate that with concerns about food availability for salmon. So next slide, Darcy. Hope you're not hearing my screaming child in the background. So we are having really long and robust conversations around adaptation. Obviously, I don't have time to get into that now, so I just wanted to relay maybe some highlights. Fishermen are adapting. They may not call it that. They may call it more responding. And that is, I think, in part because their baseline is really around adaptation, right? Fishermen are always sort of figuring out ways to change their systems to adapt to different things that are happening. So changing, sizes in their mesh sizes in their nets, where they're fishing, diversifying their income streams, mariculture, and so on. But many obstacles stand in the way of them adapting and building further resilience into the future. So we have conversations around these various sort of entities that are getting maybe in the way of building this broader resilience. And since this is a science group, I thought I would sort of note that the issues around attribution and science that come as part of the sort of portfolio of obstacles in the way for fishermen, meaning that they discuss how scientific complexity and uncertainty around climate change makes it difficult for them to conceptualize whether ecosystem changes that they are seeing on the water are related to climate change, and therefore whether they are something that they need to ultimately adapt to. I think OA may be sort of a science communication success story in that realm, again, because it seems to cross these sort of broader socio-cultural, political boundaries around climate change attribution. Again, in that even for those who don't necessarily believe climate change is happening, they are still sort of believing in ocean acidification, which, we may sort of have something to learn in terms of climate change communication more broadly from the OA community. And then some areas for further improvement that fishermen have talked about in terms of relaying science and building communication networks between fishermen and scientists around climate change. And those are sort of, around developing cooperative research and communicating directly with fishermen, providing the science in an accessible way. And also, I think, interestingly, leaning on explaining the present as opposed to, forecasting the future. I think a lot of fishermen sort of are wary of our capacity to predict the future, but they would like information about the ecosystem changes that they are seeing and how they might be relating to broader climate change processes. So that's it. I know I went over my 5 minutes, but thanks for the opportunity.
Thanks, Marisa. That's really fascinating, and I think really important for us through the research community to think about our fishermen audience and what they are receptive to and what they need more of. And I think we will be able to dive into some of these topics in more detail during the OA discussion series that will take place this spring. So thank you I know even with preliminary results, it's really helpful for us to hear those. I think our last speaker today will circle back to Chris Long, who I think is now able to connect with a better audio connection.
Okay, can you hear me?
Yes.
Yay. All right. I'm going to blame the NOAA thing myself. It's NOAA and Alaska connection. I think that's the fatal combination there. So, all right. So as I was saying, we, you know, this kind of 3-year funding cycle, which we're in the second year of, we've really been kind of moving towards multiple stressors or factor studies and towards trying to understand mechanisms a little bit better, both through physiology and through gene expression type studies. So some of the stuff that we got done in 2021, which I really have to put a big thank you to just all the people who worked on this during the time of COVID, reduced staffing allowed in the lab and all of that stuff. It took a lot of coordination, a lot of work. Yes, sorry, forgot to mention who I am. I work in Kodiak at the Alaska Fisheries Science Center out here. So anyway, we've been studying crabs here since 2007, I believe, and their effects of ocean acidification. So some of the experiments that hopefully getting finished up this year. I was actually supposed to finish the first one tomorrow, except I decided to get COVID instead. Just the effect of food quality across with ocean acidification on red king crab juveniles. We've seen a negative effect. We've seen that they don't seem to have the ability to increase their food ration, and we're wanting to kind of figure out how much of this is an energetics thing for them. So we've been feeding them a variety of diets in addition to exposing them to OA in this one. So that one, like I said, hopefully will be ending in the next few weeks. We've done a first look at snow crab juveniles, just at 3 pHs, looking at growth and morphology. That one is currently in progress, and again should hopefully be finishing up within the next month here. We're waiting to get one more molt out of those guys before we end it. We completed a study looking at gene expression in snow crab adults. This is the lower picture in the panel here. Those samples got taken and they've been sent off for mRNA analysis. So those are exciting forthcoming results. And then following up, we did a study a few years ago with papers that are in review right now, looking at how ocean acidification affects embryos and larvae in snow crab. And even with a 2-year exposure, we found no effect on either of those life history stages. And so we're doing another experiment where we're looking at this again, but we're crossing it with temperature this time and seeing if temperature affects it. We have just the high temperature treatment, but also how the combination will. So we're about 1 year into that experiment right now. The first female is just starting to hatch her larvae into that one. This overlaps a lot with what Andie was talking about earlier, but this is the OA monitoring we're involved with in Kodiak. The percolator is a machine you guys are probably familiar with, but it sits in our laboratory, and we got continuous data for most of 2021. We were able to get back in the late winter to start that machine up again and get that data stream going. And fingers crossed, so far we haven't had to turn it off this year yet. So we're hoping to get that that beautiful series of graphs that Jacqueline showed us just repeated for the Kodiak area here. And then with our KANA partners, the remote site monitoring project, again, like Andie said, that was off to a rough start, and COVID just threw a lot of wrenches in at that one. So we've been getting that one started back up again this year, and we're hoping to get a good year worth of data out of that. As we move into 2022, we're going to be continuing to look at different life history stages and different factors. We've started some behavioral work with crabs. We've done some work with juveniles, and Aliyah Zacher, who's one of the researchers up here, is going to be looking at larval behavior under different pHs. we had Shelby Bacus is going to be coming out here, and she shared about these next two projects, so I won't belabor them, but just looking at cross effects of ocean acidification and temperature on snow crabs and counter crabs, and being able to compare those two. We're planning to do an OA selection experiment. That's where one, I'm actually very excited about it, where we'll hold tanner crabs in the lab. They're one of the most sensitive species we've found. And we'll hold them until about half of them die in the acidic treatment. And then we'll do full genome sequencing on those as well as control crabs and look for markers of selection. And that's going to give us some really interesting information about which genes are selected for, and we'll be able to compare that hopefully to some of our gene expression experiments and better understand the mechanism. And then finally, be looking at hemolymph, that's crab blood, chemistry in tanner and snow crabs, and how that changes over time after exposure to low pH. And lots of thank yous. OARC are the owners of our percolator. They've had it on loan for us. OAT funds us, and we have a whole lot of people who work in the lab to keep us running. And any questions?
Questions for Chris? Chris, thank you for giving your presentation despite dealing with COVID. Extra points for you.
Yeah. It's our second go around as a family. It's Omicron is a lot less than Delta. It's only slightly, the Omicron was only slightly worse than the second COVID vaccine for me, but.
Oh, well, good energy to you and your family. Anybody have a question for Chris? Okay, let's see, Chris, if you discontinue screen sharing for a second. Great. I guess I first say, is there anybody else online that would like to give an update that didn't have a chance already? Okay, I think we made it through our program, and I just want to take a moment and acknowledge that you all just represented an incredible amount of research, and it's really heartening to see all of this moving forward. I feel like, as we all know, ocean acidification is a hard topic and it's a depressing topic, and we have such a vibrant research community here, and everybody on this call is making a difference, and it feels really like a lot of wind in our sails to be part of this community. I think our challenge that we have many challenges, but one of them Jessica highlighted is making sure that the relevant and timely information gets out when it's ready to the people that need this information. And we will continue to try to get people's attention on this topic so that we can make headway on emissions reduction and carbon dioxide removal, which is, of course, I think is the ultimate goal through all these studies, is to try to slow down this problem and adapt. I have kind of a silly question, but I was thinking that if those of us that are still on the call, if we turned on our cameras and took a quick picture, a COVID community ocean acidification picture, that might be kind of fun. And I might need help from someone else. I transferred into a different room of my house because my children have arrived and are now getting a German tutor lesson. So I'm on my little screen, and I'm wondering if someone else here could take a big screen photo of everybody. Oh, look at you all. It's fun to see the names turn into all these real people. So maybe we'll say on the count of 3, everybody smile. 1, 2, 3. Okay, hopefully a couple of you took some screenshots. Awesome. Okay, I have a couple more short requests, and one is if everyone could send me their slides. Some of you have really cool graphs or take homes, and when I'm putting together presentations for public audiences on behalf of the network, it's really helpful to have some of your most recent information, and I won't use any of your graphs or slides without checking in with you first, but it's really helpful to have them on hand to make sure that I am conveying accurate information. Also keep an eye out for the recording from this session. We'll circulate with everyone and also post it on the Ocean Acidification Network website. And then lastly, enjoy the Alaska Marine Science Symposium. Check out that long list of ocean acidification talks. And we hope that you will join us for the ocean acidification discussion series this spring. So with that, have a great evening, everybody, and we'll be in touch.